So I want to introduce to you Hubert Bauer, who graduated from his studies in biology in Würzburg in Germany in 2009. And after working as a scientist at the same university in 2019, he became member of the board of applied genetics in companion animals. And to his professional career in 2014, he joined LaboClean's microbiology department and focuses on the interface between the genetic science and the customer, which are often breeders. So Hubert Bauer, very glad that you are with us, and the stage is all yours.
Thank you so much. I hope the mic is taking up well, and then I share the screen to the slides. Everyone hears and sees.
Wonderful.
And I'm very honored to be able to present a little bit of genetics as well, since it's a field where a lot of technical advances and also scientific advances allow new tools for our customers, the breeders and breeding clubs, as well as for vets, to see what is actually happening in their breeding populations. So as a start, I put some of the let's see. I put some dogs on our grass in front of our company, so they look almost like genetic clones. And indeed, that's something to talk about, the genetic loss of variety in companion animals and especially in dog breeds. When we talk about genetics, just to bring everyone on the same level again, it's about to talk about the inheritance of the alleles. There is always a father and a mother which pass one of their two genetic copies to the next generation. And therefore, the statistics behind that and the available variation in markers and combinations available also on a population level are very relevant to take care of. So a beautiful pic illustrates that in addition. Here we have a lot of German Spitz sitting next to each other, and there is just one genetic loci involved. The intensity gene for the coat color yellow shows that much of variety within a single breed. So this beautiful diversity is to maintain, and also, of course, on the whole genetic level for the breed itself. Just historically, dog populations have been undergoing, of course, various bottlenecks, which have been devaluing the genetics behind it. Most obvious at the point where the wolf became domesticated. There has been only so many of individuals being relevant for the dog populations. And therefore, from a broad pattern of variety in the genes, this leads then also over the time of generating a breed, selecting dogs for special purposes, for special phenotypes, to a pattern in genetics which is quite even, quite equal, which shows only big similar regions. And these big similar regions are once, of course, relevant to make the dog the breed it is, but also are relevant if you track inherited diseases. On a population level, this is what happens when you set bottlenecks to a population. The ideal population is always the one which undergoes slow growth and allows a lot of genetic diversity. With some bottlenecks present, there will be genetic diminishing of genetic diversity and deleterious variants rising in the same matter. In addition to that, we have to consider how dog breeding works. There is often the case that the winner of a show, the popular one, the best in show, is the champion of the breed and therefore inherits way too much of its genes to the next generation of dogs. Nevertheless, those popular sires, often been used in the last decades or even now, are still the individuals which shift the genetics of a dog breed to a certain direction. And this is not necessarily always the best one, because with showing up good, he still can contain some sort of genetic burden, a shadow. Even if every single available genetic test is done, those dogs might carry maybe a complex polygenic disease, which is not known yet, or some alleles which might be deleterious and are not known to science. Therefore, we have to consider that the drift those popular sires introduced to the population even accelerates what's happening to inbreeding depression in dog populations anyway. The genetic drift is something we have all to take care about. So just leading from an equally distribution in initial generation, then a marker gets ubiquitously distributed into a homozygous marker over time, just through the sheer effort of passing genes from one into the next generation. It's something which happens automatically, and of course not within 3 generations, but in meanwhile, the dog breeds reach the actually relevant generation times. 30, 50, 100 generations of close dog breeding are already occurring, and therefore the genetic fitness of a breed, the reduction of genetic diversity and the loss of heterozygosity is happening. Therefore, some science has been done already. There is impact on litter size and longevity described, and it's a challenge for the dog world to counteract that. Then there is even additional selective traits. Of course, science progresses, and meanwhile, I was just putting that screenshot from the website of the inherited diseases 2 days ago. This number always rises. There is meanwhile 850 different traits known just for the dog. And yeah, during the next presentation to that topic, there will be over 850 for sure. And in addition, of course, there will be always unknown variants, and also science does not very well understand polygenic traits in the companion animal world yet. Also, complex diseases with multiple factors combined are relevant to cause genetic disease. So all of this combined is something which tackles the selection of the proper breeding partners. And it's not only always a disease, it's sometimes just susceptibility to a drug, which is also relevant. I brought with me the example of the multi-drug resistance gene. There is a defect or a mutation occurring in some shepherd breeds, which is quite of high frequency. Just to find a collie with an allelic frequency of over 50% in the breed as a breeding partner is challenging, because there is other inherited diseases which makes it hard to even combine dogs for a mating without causing deleterious alleles to meet in a combination that no sick dogs are born. Altogether, we have been learning that this homozygosity and selection has been taken part over the last generations for the dog world already. So selecting the phenotype is always more important, or selecting for behavior is more important for a breeder sometimes than just to see if there is something maybe occurring in loss of genetics. So this has been shown in some publications. I brought some examples with me. For example, we know that homozygosity is actually basically relevant on a genetic level, even to show some behavioral things. The pointing behavior for this Weimaraner dog, for example, has been studied intensively. And here you see those homozygous areas within the pointing dog breed. This is limiting their genetic variability, and then, of course, accumulating maybe disease factors in the breed. So how to counteract that? How can we react on that things happening? As a geneticist, I would answer, oh yeah, we have a tool. We can do population-wide breeding programs. And this is what the livestock is basically doing. If there is certain selective traits, you can combine a good one and a bad one. You can make a good data acquisition of the whole breeding population and actually tell the breeders how to contradict the effects taking part in the population. Unfortunately, this is something with a whole breeding program, with a selection of traits, data acquisition, selection of markers within a big array of data, or giving a breeding score, and then actually breeding after a breeding score, and a re-evaluation over time. This is something only big breeding programs can accomplish. And dog breeding is often something occurring on a little lower level than this, which can be done by the big breeding companies for livestock, where the money is basically talking, and the liter of milk gained by this kind of breeding programs is actually relevant for each dollar earned. In the dog world, breeding happens how the breeder wants to do it. The fun of breeding for a breeder is to put his opinion on the next breeding partner he selects. And he will most likely not care about a breeding score given by a geneticist. So it is to actually acknowledge which strategies are left to maintain and maybe promote diversity for the individual dog breed. What is happening still way too often is narrow breeding or line breeding in dog populations, which is actually diminishing the diversity of the individual and accumulates recessive disorders, also raising inbreeding depression. And what is the tool? What is to select is our dogs which are genetically most distant to the breeding partner you want to breed. The outbreeding strategy, to search within the population and therefore find new combinations of genes which have not been even present maybe before. This raises the diversity of the individual. On the population level, this has no effect neither, because there is no new genes introduced, of course. But at least we can diminish the accumulation of recessive traits and have an effect on inbreeding depression for the line bred. Then with some of the outcrossing events are also happening. If you indeed look over the border of your breed, you can find a similar, genetically related, maybe similar looking breed with new lines and new genes. Unfortunately, these events are very rare. So there is a couple examples from Northern European countries or in America where there was no other option anymore than to breed in between the borders of a breed to boost their own genetic again. But this is something which is basically the last hope for a breed. So outbreeding maintains as the strategy. And of course, this is already done just by classical approaches in the breeding clubs. Just by sheer pedigree analysis, for example, calculating an inbreeding coefficient there, and maybe also ancestral loss for the dogs, can give a good hint, a good base to find genetically distant individuals as well. But it requires, of course, the effective the pedigree over 5 or more generations back. Better if it's 10 and more generations back. And this is hardly available, especially in a field where breeding occurs more international over time. There is possibly an ideal breeding partner, it seems, from another country, and I have no information about his pedigree and how he could be related to my dogs. And there is where we come in as a laboratory providing genetic tests. Genetic data can gladly and greatly help in that field to actually monitor the heterozygosity index and then give an information about the genetic diversity of the different dogs measured. So the monitoring of those dog breeds is needed, and the technical advances are giving us no headaches anymore. the simultaneous recording of many data points over single nucleotide polymorphism, the SNPs, is readily available and easy to do in a large scale through next generation sequencing or microarray studies. And then there is only the challenges which remain to get all relevant data. So if you want to ask for population genetics, you have to at least try to get all lines and most of the breeding individuals of a breeding club to get an overview of what is present in the genes. And since you have to get the samples from the breeder and the breeding clubs, you need to find a clear and integrative communication with breeders and breeding associations. A great help there is the International Partnership for Dogs, where a lot of the big European clubs and also the American Kennel Club meets together with labs providing those kind of analysis, and also the veterinarians and scientists in the field come in and give their guidance and counselation for the clubs to act upon the things happening. So it has been of great value for us to be in that group and to then understand how we can deliver low barriers to breeders and breeding clubs to join into that process of letting us analyze their populations. The tool of choice there is the DNA profiling test. Breeders are basically used to the fact that they have to check on parentage and on the ancestry of their dogs. So a DNA profile, a genetic fingerprint, is already available since over 10 years, and based on a 22 marker set, parentage is something they have to do anyways already. With technical advances, the standard set to analyze those DNA profiles has been changed towards 230 SNP markers now, and all the machines running those analyses basically have an overshoot of information, so you can easily run a couple hundred, a couple thousand markers more for every individual, and therefore not only do the parentage check, but also check for relatedness or do those analyses which are needed to find out heterozygosity status and the genetic diversity of the dog. And then finally, coming to some of our data, which has been generated in collaboration with breeding clubs. Of course, just looking at the heterozygosity status of breeds is already helping a little bit how severe or how intensive the genetic loss has been happening over time in that breed. Here compared a West Highland White Terrier to a Golden Retriever, just on the sheer base of the distribution of the heterozygosity in percent, will tell you the Golden Retrievers have a way bigger breeding base as the West Highland White Terriers. And then looking at the numbers, the maximum here at 25 is way lower than the maximum at around 30 for the Golden Retrievers. That's quite a difference already. And it simply explains, of course, through the little or smaller breeding population of West Highland White Terriers. There you might actually indeed want to change and shift a little towards more heterozygosity within the breed. And the customer always gets a data point where his dog is compared to the rest of the population, which is the background. So he can already see if there is maybe a dog which is actually very fine and is more heterozygous than the average of the breed, or if he might do some changes to his line if he's left from the middle and therefore maybe needs a little bit of a genetic boost. But going into detail is something we have to assure as well and allow to find a possible breeding partner. And there we chose this analysis, a principal coordinate.
30 seconds left. 30 seconds left, Hubert. We need to conclude.
I will speed up. This is the basic, the relevant graph. We have principal coordinates analysis available where you see actually the distribution in between the dogs of the breed. Some lines might be highly accumulated. Of course, this is equal to what happens in the inbreeding coefficients. You will have closely related dogs in a dot, but then you can find your breeding partners and outbreed to the rare alleles of the breed. This is the main tool used by breeding clubs right now. Here, a data set shown from 2021 and 2022, some project we started already 2 years ago with a Parson Russell Terrier Club, and now is readily available in an online platform and can be used to combine those strategies. Classical genetic tests are relevant, but now with the new possibilities, we can preserve heterozygosity for the breed. We can prevent inbreeding depression and have those genetic tools readily available for our customers. So thank you for your time and your attention. Let's all together maintain that high levels of diversity also for the companion animal sector.